A person's body contains millions of cells, and they all have a specific function. Muscle cells allow the body to produce force for movement, while nerve cells transmit signals from the brain to other parts of the body. But where do all of these cells get the instructions to perform their functions? All of a person's genetic information is coded in their DNA. The DNA is essentially the blueprint that spells out the specific function each cell has to perform. Without a specific set of instructions from the DNA, the cells cannot carry out their functions. Since all cells will eventually die one day, it is important for each cell to reproduce. But the new cells created must have a set of blueprints of their own so they know how to perform their specific functions. This is one of the most important reasons why DNA replication is a crucial process for cell survival. Without properly replicating their DNA, cells cannot undergo cell division and grow into two new cells. Thus, the overall message of this educational video is to describe and graphically illustrate the process of DNA replication and its important role as a part of cell division. The DNA holds all of its genetic information in the nucleus of the cell. It's made of 4 building blocks called nucleotides: adenine A, thymine T, cytosine C, and guanine G. These building blocks have a special connection in that A always pairs with T, and G always pairs with C. In addition, the DNA is made of 2 strands that wind together to form a helix structure. Both strands have a 3' and a 5' end, and these markers drive the direction of replication. To explain DNA replication, I'd like you to imagine a cell as a town. It's a bustling metropolis with a lot of commotion and traffic. It's getting so big after a long growing period that the mayor of the town wants to expand. But the problem is that there is only one copy of DNA. The DNA that is inside of the town hall, or the nucleus, has all of the blueprints to map out the construction work plans. But with only one copy, it's not enough for all of the workers to have the information. So the bottom line is that for the cell to divide, we need to replicate the DNA. To start the replication process, the mayor hired a special team that specializes in replicating the DNA. This special task force consists of mainly DNA polymerase, helicase, ligase, and RNA primase. They work for the company Enzymes. To begin replication, helicase attaches to the DNA at the origin of replication. It pries the DNA open by breaking the hydrogen bonds between the nucleotides. In addition, he uses special gadgets called single-strand binding proteins. These little guys hold the double strand open so that 2 single strands are revealed and act as templates. This process continues down the line as helicase unzips the DNA. Next, to add on new nucleotides, RNA primase adds a short stretch of RNA called primers. These primers allow DNA polymerase to add nucleotides to the template strands. Before getting any further, there are some important information that must be noted. Nucleotides can be only added from the 3' to 5' ends by DNA polymerase. But since the 2 strands of templates are asymmetrical, meaning the 3' to 5' run in opposite directions, there will be a leading and a lagging strand. The leading strand will have nucleotides added continuously as you move towards the replication fork. However, the lagging strand has to constantly add small sections of nucleotides called Okazaki fragments. Each time an Okazaki fragment is added, it must be primed by RNA primase and have DNA polymerase add the nucleotides. Finally, ligase comes in and connects the new fragments with the previous ones. Overall, as you constantly move to the right, the helicase unzips the double strand. The leading strand quickly gets nucleotides added, and the lagging strand slowly catches up. On the left side of the origin, the same thing happens, but notice the flip-flop of the leading and lagging strand. But this makes sense when you notice the direction of the 3 to 5' on the left side. After a long day at work, the Special Task Force finishes the replication process. They are well thanked by the mayor, and the workers are happy to get on their construction since they now have the blueprints to expand the town. In the end, the original long double-stranded DNA becomes two identical copies. With the DNA replicated, the cell can now move on and start the cell division process.